All right, welcome to Lecture 11. We're going to be on Plate 48, starting the structure of the respiratory tract. It's all going to come down to oxygen and CO2. We are aerobic organisms. We live in an air environment where oxygen 50 times richer than in an aquatic environment. We are still faced with the problem of getting rid of CO2, which is a waste product. Now that I've said that, we also not only regulate the concentrations of oxygen in our body, but also regulate concentrations of CO2. But what I'd like to do now is start off with just the basic anatomy. On Plate 48, it shows a human. So this is the human view. If you want the view of the dog, just turn it 90 degrees. The elements, the anatomical elements, are very, very similar. So let's start with the conducting passage. We start with the nares, which are the nose openings. They are under muscular control. You see people get angry, the nostrils flare. Horses, the increase, that nasal flaring is critically important in increasing the tube diameter to allow air entry in to the nasal sinuses. In the nasal sinuses, air is humidified and temperature is increased. From the nasal sinuses, air passes into a large common vault called the pharynx. The walls of the pharynx are lined with lymphoid tissue, so this is one of the lines of defense when air comes in carrying microbes and extraneous organisms. The pharynx is the site of presentation to the immune system. It's also a common vault for food transfer. And therein you have a bit of a problem. If you look in a quadruped that's laying down, here's the pharynx, so air is coming in this way. Food is coming in through the palate and through the mouth. It's pushed back, and the food has to go up to the dorsal esophagus. The air must go down ventrally to the trachea. So you see here is this common area, this pharynx. So there are protections to prevent the movement of food into the trachea, and that lies with the larynx. The larynx is a muscular and cartilaginous structure that guards the entrance to the trachea. It has around it the glottis and epiglottis. Some of these structures you'll be looking at in lab. Air moves from the larynx into the trachea, also invested with smooth musculature and cartilage. It's a rigid structure that branches into two bronchi. So there's a left and a right bronchi. The bronchi branch, much like the arterial system, into smaller tubes called bronchioles. The bronchioles feed into gas exchange structures called the alveoli. Most of this that I've talked about, most of this transport ductwork, is not involved in exchange. Gas exchange occurs at the alveoli. So any air coming in has to go through all of this space and then return through that same space. A process that is called tidal ventilation. Fish, as they swim through the water, pass the water media with the oxygen into the mouth and out through the operculum. So it's a one-way flow. Birds have done a very nice job in anatomically structuring so that air kind of flows one way through their lungs. But mammals are faced with having one set of ducting, the pharynx, larynx, trachea, bronchi, and bronchioles, and any air that enters the alveoli must exit through those same structures. A hallmark to gas exchange is a thin cell membrane. So if we look at the bottom of our plate, that's exactly what we see right here. This space is represented of the gas interface, so this is the alveoli here. Here is a capillary which is feeding the lungs. So these are referred to as pulmonic capillary. There are the red blood cells, deformed as they squeeze through. That deformation allows for close approximation of those red cells to the cell lining of the capillary. Very small interstitial space. That is going to reduce the distance for diffusion. Thin endothelial walls of the capillaries, thin interstitial space, and very thin lining, alveolar lining. What isn't evident in this figure is a thin boundary of fluid. You don't want to have too much fluid, then you would be drowning. But there is a very thin layer of fluid which lines the inside of the alveoli. That means gas in air must first then dissolve into the fluid, diffuse through this fluid, then diffuse across the endothelium of the alveoli, through the interstitium, and through the endothelium of the capillaries, and finally into the red blood cells. Plate 49. We visited this anatomy when we were talking about the cardiovascular system, but let's revisit it again here. The lungs are confined to the thoracic cavity. The thoracic cavity is defined by a rigid structure consisting of ribs and muscles. These muscles fit between the ribs. Latin for ribs is costal, so there are intercostal muscles. There are muscles that are on the external aspect and the internal aspect of these ribs. So there, in fact, are external intercostal and internal intercostal muscles involved in the breathing process. The posterior aspect of the thorax is defined by this thick sheet of skeletal muscle, the diaphragm. The thorax is a closed space. The lungs that sit in the thorax are connected to the external environment through the bronchi and through the trachea. So these alveoli are in contact with the external environment, but the inside between the alveoli and between the interlining of the thorax is an enclosed space. This is referred to as the pleural space. Under normal circumstances, the pleural space is always under a slight negative pressure. We have horses that run through the field. They look for sticks. If they can't stick it into their eye, they try to stick it into their chest. If you have an insult to the thorax, an opening into this thoracic cavity, you now allow access of external environment to the pleural space. If the pleural space is slightly negative, air rushes into the pleura and the lungs collapse. This is referred to as a pneumothorax. Air in the thorax And the problem with this is that when you inspire, instead of air coming down the trachea and into the lungs, it gets pulled through this hole. As a consequence, the lungs don't expand. I just got a better tan. You notice that? Okay. So air comes into the thorax and not into the alveoli. This is demonstrated at the bottom of plate 49. Respiratory pressures. There's intrapleural pressure, so that's the pressure within the pleural cavity. Intrapulmonary or alveolar pressure, so that's the pressure here. Normal atmospheric pressure, that's the pressure in the external environment. So these numbers are all in reference to pleural pressure, pulmonary pressures, alveolar pressures, and the external environment. So in rest, so I want you to just sit there, don't breathe, just keep your mouth open. Your thorax is neither expanding nor contracting. At this point, the pressure inside the alveoli, or pulmonary pressure, is identical to the atmospheric. Pleural pressure is still slightly negative. Now what you're going to do is you're going to inspire. Inspire uses the external intercostals and the diaphragm to increase the volume of the pleural cavity. Because we have a closed system, if you increase the volume, that will decrease the pressure in that. And so you see you go from -3 to -5. It's not a huge pressure change, but one that sets it now apart from pulmonary pressure, alveolar pressure. Because the lung is elastic, and this is a critical component. If the lung is not elastic, if it's a solid structure, it's uninfluenced by pleural pressure. But because it's like a balloon, the walls of the alveoli expand, and the pressure in that also drops. So it goes from 0 to -1. Now you have established a pressure differential between atmospheric and pulmonic pressures that will force air into the alveoli. At the end of inspiration, okay, so you've stopped inspiring, your thoracic cavity is still larger, but at this point, you've equilibrated. Pressure in the alveoli and pressure in the atmosphere now are equal. And then what happens is you expire. Expiration is usually a passive process. It involves relaxing the diaphragm. And in active expiration, you invoke the internal intercostal muscles. So if I asked you to force expire, you could do that, right? You contract those the internal intercostals. In either event, what that's going to do is you're decreasing the volume on inspiration. That's going to increase the pressure, and you see that it goes from -6 to -5. Now you've established a pressure to force air out of the alveoli. Airway resistance. We've talked about fluid flow, fluid dynamics, the physics of blood flow. Physicists view air and water similarly. They are both fluids. They just have different densities, but they are under the same physics and regulation of physics. One of the things I really jumped up and down about in terms of cardiovascular flow was that blood flow was a function of the pressure differences and the resistance. In the cardiovascular system, we were talking about pressures of hundreds of millimeters of mercury. In the pulmonic system, we're talking about pressures that are an order of magnitude lower. Well, that's because the viscosity of air is a magnitude of lower than fluid. So you can have huge flows of air with little changes in pressure. So that's the first difference. The second difference is, I said second difference. How about a second important component is that airflow is under the same constraints as blood flow in terms of resistance. We said that viscosity was one of the huge differences between air and water. Both are influenced by the radius of the tubes, and specifically radius to the fourth. So in the same fashion that if you increase the radius of a blood vessel by 2 times, it would lead to an increase in blood flow of 16 times. That's identical to the conditions with airflow. If you were to increase the radius of the bronchi by 2, for the same changes in pressures, you could increase airflow by 16 times. And in fact, that's one of the ways we're going to change. Now I've gone to the bronze look. What did I do? Okay. All right. So let's turn to plate 50. And look at surfactant, surface tension, and lung compliance. So you knew we'd come back to compliance. The pen? Oh, what am I? Did I hit the wrong? I bet I hit the wrong thing. There we go. We talked about compliance of the veins. We said that veins were very compliant. Now what I'd like to do is talk about compliance of the airways, and specifically the lungs' elastic behavior. On the top left of plate 50, you see a plot of volume versus pressure. This is the lung in two, so let's look at an air-filled lung. So the air-filled means if you change pressure, there's this change in volume. Change in volume over change in pressure. But what's happening is, as you change the pressure in the interpleural case, there is a tremendous change in volume. If you fill them with water, it's much easier to change the volume. So what's happening? There is an interface in the air-filled lungs between water and air. There's a thin layer of water on the alveoli. A layer of water that resists changes in shape. It is those hydrogen bonding of the hydrophobic and hydrophilic ends that produces this tension or resistance to movement. That is what is referred to as surface tension. We need to have elastic lungs. The alveoli have elastic elements in it, but they are faced with this formidable problem of you have this thin layer of water, and this thin layer of water is going to resist changes. It's going to decrease the elasticity. And how do you deal with that? You add in a soap that disturbs the surface tension. Now, you do this every time at home when you add a little ivory soap into the dishwasher, is you alter the surface tension of water. It allows for the water to be less cohesive, less adhesive, and to release dirt and particles. In the lungs, you're seeing a very similar thing. You put this lipid, soap-like structure, that starts interfering with the bonding of the water molecules. So this surface tension, this resistance to expansion, is destroyed or reduced. That compound, that lipid compound, is called surfactant. Surfactant, surface active ingredient. It is a lipid moiety. Its presence increases the elasticity of the lungs by about 2/3. So not only is there elastic elements of the alveoli, there is the presence of surfactant. What does that reduction in surface tension mean to the lungs? Well, first of all, it makes the lungs more compliant. It's much easier to increase the volume of the lungs. So small changes in pressure. If you did not have surfactant, you would have to generate much higher pressures to breathe. The second element that surfactant plays is in reducing the surface tension is allowing different size alveoli to exist. So let's look at effective bubble size, tension, and pressure. So here's a bubble. You can think of this as an alveoli, kind of a circular shaped structure, and there's an alveolar duct coming into it, so air is coming into it. The pressure that's needed to keep this thing inflated is going to be a function of the tension on the wall divided by the radius. And that the fact that the radius is in the denominator is going to mean that small bubbles are going to be in a tougher situation than a large bubble. Or you have two alveoli that are connected, there is going to be a tendency for one of those bubbles to collapse and the other one to expand. That's demonstrated down here. Large bubble, so the radius is much larger in this. They've designed it so the radius of this one is 1 and the radius of this one is 2, so there's twice the diameter. Pressure, and arbitrarily we put pressures of 40 in here and 20 in here because 2 times 20 over 1 versus 2 times 20, so that's tension, and these could be millimeters of mercury, tension over radius, and it's 2 times. Well, with twice the amount pressure in this, air will flow from this bubble into this bubble. That will result in a collapse of this bubble. With surfactant, tension, surface tension, is reduced. So one of the numerator values drop. And you don't have to remember that it's 2. You just need to know that the pressures that are required to keep these bubbles open is a function of the surface tension divided by the radius. And I won't ask you to do math in this at all. but to understand that if you have two bubbles, one small, one large, they are unstable. The tendency is for this bubble to collapse into this bubble. And you can see that at home. You've got soap bubbles. And you watch. Little tiny bubbles will do what? They will come together, they will coalesce, and form a larger bubble because it's inherently more stable. Well, the problem in lungs is you want to keep the alveoli small. That increases the surface area. Without surfactant, the alveoli tend to collapse into larger, reducing the surface area, and is one of the problems with an infant respiratory distress syndrome. The original work in these infants, they were born without surfactant. They were having trouble getting air into the lungs. And the initial treatment, in fact, involved taking exudate from calf lungs and spraying it by an inhaler into the nasal passages of the human infants. They would inspire this, spread this surfactant along their lungs, and all of a sudden be able to breathe. So without surfactant, alveoli would collapse. So we've got a functioning lung. We've got the anatomical setup. Now let's go ahead and see if we can do lung volumes and ventilations. I may have to go to another plate. I think so. That was plate 51. My next improvement will be to have these all just pop up at the right size. So let's start here with normal inspiration. So this is the resting situation, non-exercised. There is a period of inspiration followed by a period of expiration. This is a graph representing those volumes that are exchanged during inspiration and expiration. The numbers are germane for humans, not for dogs. So I don't want you to know the volumes. What I want you to have a concept of are the relative volumes. And I'm going to give you some numbers just so you can kind of see the magnitude of these changes. In a resting human, every breath you take in takes in about 500 mils. And that would be represented by a move in this direction. So there's an inspiration of 500 mils. When you expire about 500 mils, depending on your gas exchange rate. This pattern of inspiration and expiration reflects a volume which is referred to as a tidal volume. Tidal because the movement of air in follows the exact same pathway as expiration. So the tidal volume at rest in a human is around 500 mils. What happens during exercise? If I ask you to increase the volume that you breathe, you can voluntarily maximally inspire. So there is a volume then in reserve for this inspiration. So that would be inspiratory reserve volume. And in humans, it's 5 times that of the tidal volume. So you can see the magnitude of these. So you've got a lot of inspiratory reserve. That means you have a lot of capacity to increase your ventilation, both for inspiration and for expiration. The inspiratory reserve is substantially larger than the expiratory reserve. You have an intuitive feel for that. If I ask you to maximally inspire and expire, you have a sensation that it's much larger volume during inspiration. And then if I actually ask you to maximally expire, trying to get all that air out past normal tidal volume is more challenging. However, during maximal exercise, you are tapping into that inspiratory reserve and expiratory reserve in what is called a vital capacity. Old term. Vital capacity, then, is the total amount of air that you can exchange, including both the inspiratory capacity and the expiratory capacity. Because the lung, the pleural space, is always net negative, there is always some distension of the alveoli, so there is what is called a residual volume. And in humans, it's about 1 liter. So that's a volume that you could never get rid of. There is not a complete and even exchange of gases. One, because you always have this air that you can't get rid of. That means that's air that I cannot totally replenish. And secondly, there is anatomic dead space. Anatomic dead space is space which is non-exchange. You can't have oxygen and CO2 exchange. That exchange only occurs at the alveoli. All of these connecting ducts have a minimal amount of exchange. That means that air that's sitting in the pharynx, larynx, trachea, and bronchi are not in exchange. They are in what is referred to as the anatomic dead space. In humans, there's about 150 mils of this dead space. That means when you inspire 500 mils, there's 150 mils that never gets down to see any kind of gas exchange. We refer to tidal volume times respiratory frequency as minute volume. That is a measure then of pulmonary gas exchange. Pulmonary ventilation. Tidal volume, the volume of air that comes in with each breath, times your respiratory frequency is the minute volume. So at rest, if it's 500 mils and you're breathing 12 times a minute, that's 12 times 500, your minute volume. Because you have anatomic dead space, not all of that minute volume is in exchange. So we have another term which is referred to as alveolar ventilation. Alveolar ventilation is the amount of air that is exchanging at the alveoli. That is equal to the tidal volume minus anatomic dead space times frequency. And that's reflected. So there's 500 minus 150. So the volume of air that's going into the alveoli is 350. So your alveolar ventilation is 350 times 12, not 500. Now that has significance in terms of if you're artificially ventilating for an individual or an animal, got an animal on a surgical table, you need to make sure that you are adequately ventilating the alveoli, not just the overall situation. If you've got a giraffe on a table, it becomes even more obvious. One of the sequelae to this, and how animals might use it, is the case of the dog. If I ask you to increase your ventilatory frequency and the volume, and you're in a resting situation, what you get is a nice buzz. Okay? You get lightheaded. And we'll talk about ventilation and respiratory frequencies in a bit. But consider the dog who's very hot. Dogs don't have a particularly well-developed perspiration. They don't have a good sweating response. So the primary means of heat regulation in the dog is by panting. Well, if they increased the volume, if they elevated their pulmonary ventilation, they would get into this problem of having too much gas exchange. So what they do is they breathe very shallowly and take advantage of this anatomic dead space. They move this anatomic air in and out without altering exchange at the alveoli. So their pulmonary ventilation is relatively constant. What they are changing is the amount of air that's moving over the anatomical dead space. What that does is carries away heat, but doesn't compromise gas exchange. Okay, plate 52. Let's talk a little bit about gases in general. And why you had to suffer through Boyle's law, Charles' law in chemistry. Pressure is force. Molecules, gas molecules bouncing around exert a force. And we refer to that as a pressure of gas. Traditionally, we measure gas pressures in millimeters of mercury. And you have a feel for how much pressure that is by sitting at sea level. There is a column of gas sitting on top of your head that extends up into the upper troposphere. Gas molecules have weight, and they impact you. And we measure that as atmospheric pressure. At sea level, the combinations of gases in air produce an atmospheric pressure of 760 millimeters of mercury. That's a number you need to know from chemistry. That was standard temperature and pressure was 1 atmosphere, or 760 millimeters of mercury. In English units, I don't know what it is, 29-something inches of cubits or stones. But in the metric system, we refer to as atmospheres, pascals, or the more traditional number, 760. What gases contribute to that? In air, most gas is nitrogen. About 79% is nitrogen. 21% oxygen. Very small amounts of CO2. Normal atmospheric air, 0.03% is CO2. Very, very small. There are other gases, ozone, carbon monoxide, but helium, hydrogen, they contribute very small. Most of the air is nitrogen and oxygen. So these molecules exert this pressure of 760. If we want to determine the pressure that each contributes, that's what's called the partial pressure. And what I'd like to do is, before we get into that, let's take a break. All right, so let's go ahead and start now with Lecture 12.